Hey everyone, so we're going to do a video lecture to just kind of supplement. As you all know, I was in that car wreck, so we finished up to about this slide. Before we get started, just like a few little notes. So I extended the homework to be due until Tuesday. I think what I'm going to end up doing is on Tuesday, I'm going to open up a homework assignment for chapter 12 and 13, but I'm not going to grade it. So therefore, it's there. It's okay, for you to use as a study tool. But I don't want anybody to be stressed about trying to get that in as a grade. I know that things are, kind of hectic for everybody around this time of the year, or time of the semester. So I'll open it up. That way, you have it for questions. I'll probably put a lot of questions on there. That way, you've got them to work through if you want to. But I will basically it'll be on there. It'll give you a grade, but I'm just not going to transfer that over into the grade book. That way, it's there for you to use. It's kind of, I'm not able to really not add it as a grade into Sapling, but I manually transfer them over to Sapling. So I just won't transfer that one over, but it'll be there for you to use. All of the other stuff is up. The discussion is up. That way you can use that, kind of compliment or comment on that, and let me know what time will work for you. I did make it 4 hours, but I honestly just made it 4 hours. That way I'm available for you for 4 hours. If, say we end up early and there's a few of you that still need individual attention, I've kind of freed my schedule for that time. So once you comment on there and I've got a time, I'll post that time and location. Most likely on Wednesday, I'll talk about it in class, and I'll make sure I post the location on there soon. So I'm hoping to do it either Wednesday or Thursday. My Thursday actually just got freed up, so I've got most of Thursday available, and I put those times on there for us to meet on Thursday if Thursday ends up working better for you. If the class is split even, then I'll most likely do 2 study sessions, but it depends. If there's a few people that just need individual attention, I'd rather kind of meet with you like that instead of doing a whole nother study session, especially if it does end up being 4 hours, because that would be 8 hours total. So if it ends up not working out for the majority of the class, or even half of the class, then I'll kind of decide what to do at that point. But I'll definitely keep you posted, and I'll definitely know by Wednesday, because the discussion will close on Tuesday night. So make sure that you've posted on there. Everybody should have their exam schedule by now, or anything else that's kind of going on next week or this coming week. So make sure you post on there by Tuesday, and I will make an announcement in class on Wednesday on what time, and I'll go just honestly right after class and look for a location and post that as an announcement. So for the rest of this chapter, really what it's talking about is other ways that we can basically just kind of snap these things together. So we've talked about lipids, we've talked about triglycerides, we've talked about what a glycerol molecule is, the hydrolysis of a triglyceride into the individual glycerol and fatty acids. Remember, it's called a fatty acid because it has the long hydrocarbon chain. It can be saturated, unsaturated cis, or unsaturated trans, monounsaturated, polyunsaturated. So make sure you're familiar with all those terms. So I kind of got through this slide right here. So this is slide 12 and 13. So I got to these slides and I did answer all these questions in class. But these 2 slides right here, they're basically just strict memorization of what these points are right here. So I'll go over them real quick, but it's not much that I can really ask critical thinking wise on it. It's mostly just knowing these terms, knowing that that triglycerides provide long-term energy, and glycogen, which is a polymer of glucose. It's how the human body stores glucose molecules for later use. Knowing that triglycerides, they do provide long-term energy. Average adult male stores about 15,000 grams or 15 kilograms, right, of triglycerides in the human body. Glycogen, like I said, it's a polymer of glucose, provides short-term energy. So glucose going through glycolysis, it provides short-term energy, just kind of like if you have a sugar rush, right? So it kind of makes sense. And an average adult male stores about 3,000 grams of glycogen. It'll last about a day until your body goes into starvation mode and starts breaking down that glycogen, which is usually stored in the liver. And after that full day of, say, no eating, triglyceride stores, they last several weeks. So you can go several weeks without food, as long as you've got some triglyceride stores in you. And so, fatty acid catabolism, it's the central energy producing pathway for many cells. So catabolism, right, breaking down. So when we break down fatty acids, or we break down these triglycerides, or some of the other forms that we'll talk about here in a second, it is the central energy producing pathway for many cells. So what that really means is that cells prefer this. There's more energy that you can get out of fats than there is that you can get out of carbohydrates or even proteins. So we can actually break down proteins. And if somebody goes a very long time or they're in starvation, that's why you do see the muscles breaking down. Is because the body will prefer fats first. If it's got glucose in the blood after you just ate a meal, it'll grab that and it'll use that. But then it'll start breaking down the fats or storing them for later. But then really last resort is breaking down the muscles, right? Breaking down the muscles and the proteins and using those to put those into pathways to essentially the goal of all of this, right? Carbohydrates, proteins, fats is to produce NADH and FADH2. And those are really used for the electron transport chain, which we don't really go into detail in this class. But it's important to know that catabolism, that's the goal of it, right? To make ATP. So fatty acids, the catabolism of these things, does provide about 80% of the energy needed. It's the energy needs of the heart and liver cells. So like I said, fats are usually stored in liver. We did talk about adipose cells in class on Friday. And it does provide about 80% of the energy needs for the heart and the liver. The heart, obviously, it's very important that we have energy there. Obviously, we have to have our heart beating, right? And it provides more than double the energy of carbohydrates and proteins. So if we had like one, say, triglyceride, or even like do it like as a per gram, so it's comparable. So they provide about 9 calories, that's big C calories, per gram of fats versus say 4 calories per gram for carbohydrates and for protein. So these things are very energy rich. And I kind of like briefly went over this whole, beta oxidation thing, but it's mostly just to kind of know that for information purposes of, we break these things down basically 2 carbons at a time. And that's that's the reason that the majority of fatty acids are produced as an even number of carbons in nature. So when we're looking at digesting these things, it's important to kind of know the process of digestion. So the digestion of these triglycerides, it begins in the small intestine. So I kind of drew over on this side this figure. So you know that I'm obviously not a very good artist, but I did kind of work on this figure to hopefully make it a little bit easier to understand if you're like more of a visual learner. So food comes down this kind of makeshift esophagus thing here and goes into the stomach, right? And so once it goes past the stomach, that's when you enter the small intestines. Now, the gallbladder kind of sits right here at the entrance of the small intestines and deposits what we call bile salts. They're released from the gallbladder after a fatty meal. There's a lot of, obviously intermediary steps that tell the gallbladder, hey, I just ingested a meal that has fats in it, and we need you to release these bile salts. But really the main gist of it is fats enter into the small intestines. Gallbladder knows that, right? And deposits these bile salts into the small intestines, and they mix. When they mix with fat, they form what we call an emulsion. Now, fatty acids we talked about have a polar portion, right? The polar head, this glycerol portion, where it has the ester linkages, and then it's got the long carbon chain, which is extremely nonpolar. And we've learned that like dissolves like, and so an emulsion really is a way to dissolve something that is partially hydrophobic and partially hydrophilic. And so we form this emulsion, and it allows lipases to get in there and do their job. What lipases do, as we talked about at the beginning of this chapter, are they hydrolyze these triglycerides into water, right? So they use water, so lipases to hydrolyze them into water and fatty acids. So hydrolysis actually uses water, right? So this would actually be incorrect. So this would actually hydrolyze them, let's see, so hydrolyze them using water. So using water, hydrolysis. So you use water to break those ester linkages to basically hydrolyze them into, I believe what this should say is glycerol. And I'm gonna say, I think that's spelled correctly. So they hydrolyze them into glycerol and fatty acids. I can like save this and then upload it again under the same name, but just kind of say edit it or something like that. So once these are hydrolyzed, and it's kind of shown right here, so this beginnings part, gallbladder, bile salts, creates this emulsion, and then once they are hydrolyzed, and so this is kind of like an expanded, this arrow right here is just kind of demonstrating that it's expanding this right here. And the inside of this right here is the inside, or the inner part of the small intestines, and then out here is what we would call the outer part. We kind of go through what we call this this lumen to get out. And so within the small intestines, we take the triglyceride and we hydrolyze it into glycerol and fatty acids. Remember, it takes water to hydrolyze these ester linkages, and obviously I didn't draw the Lewis dot structure of all of this, but really what this head group is demonstrating is the polar portion, is the glycerol portion. And these long carbon chains, they're demonstrating the carbon chain or the fatty acid, which would, right here in this portion right here, the ends of these would obviously have this carboxylic acid group, right? So I've kind of drawn them as just kind of long little squiggles, but just know that those are long carbon chains, right? Each one has tetrahedral molecular shape, electron geometry. They may be unsaturated, they may be saturated. It's not really important for this part of it. So once they're hydrolyzed into the glycerol and the fatty acids, these molecules are now small enough to enter into the intestinal mucosa, where they're esterified right back into triglycerides and then packaged into these things that we call chylomicrons. And I believe that I gave like a kind of a picture of, so these kind of like are what these chylomicrons will look like. So they're a way to transport these things that have very non-polar portions of them in the blood, which is extremely aqueous, and what we know about water is it's extremely polar. So we have to have something that is able to be transported. And you can see like the polar head groups are all on the outside. So that way the water is interacting with the polar head group. And then on the inside, we have all these non-polar tails. And you can kind of see right here where my mouse is that triglycerides are packaged on the inside. So it keeps the non-polar portion of the molecule away from the water so that they're able to be transported. So they're packaged into these chylomicrons, which carry them through the lymph and circulatory system. So we have a lymphatic system and a circulatory system. You'll learn that pretty soon when you get into like more of the bigger macroscopic part of nursing. And so the circulatory system's two target cells. So we got to find a way to get these fats that we just ingested to the cells, so the cells can then use them as an energy source. So chylomicrons, they are the largest of the lipoproteins. So what is a lipoprotein? It means that they're a combination of lipids. We've talked about what lipids are. It's what this chapter is really on. And proteins, which are amino acids. And these amino acids have this core structure that we've kind of introduced as well. So they're spherical shaped. They're assemblies of these lipids and proteins. And the protein portion would be these right here. And so this protein portion is really a big part of that, is so that lipoprotein knows, once it hits the cell has receptors that grab onto those proteins and pull them into the cell, and utilize those to what we call endocytose. They bring them into the cell once they get there. So they're transported in the blood to the target cell. Then they are lipoproteins, so they're hydrophobic on the outside and hydrophilic on the outside, the portion that interacts with the blood, which is extremely aqueous and polar, and they're hydrophobic on the inside. So once it gets to the target cell, they are again hydrolyzed for transport into the cell. So they're hydrolyzed and then transported, these fatty acids are transported into the cell, and then they're converted back into triglycerides and stored or used to generate energy. So they're stored for later or used now. And so it's really just a process of hydrolysis, esterification, to get these things into the cell. And then, the final result is they are triglycerides in the cell. So it's important to know where they esterified, where are they hydrolyzed, why are they hydrolyzed, what's the importance of these chylomicrons, and the polarity of the blood versus the non-polar nature of these triglycerides. And so cholesterol and triglycerides, so we're about to talk about cholesterol too, but there are two, and I kind of copied and pasted this slide at the end of the cholesterol portion. So cholesterol is extremely nonpolar too, and cholesterol is important. We need cholesterol, right? We've always talked about cholesterol is so bad, or I'm sure you've heard cholesterol is bad, cholesterol is unhealthy. Well, it turns out we actually do need cholesterol, but too much of one thing is always not good. And so we have to have just enough cholesterol, but not too much. And so our bodies are designed to transport this cholesterol to the cell and to the cell where it uses it actually in the lipid bilayer, which we'll talk about here in just a second. So I kind of wanted to put this slide here as well. It was like placed at the end. I wanted to put this slide here as well. That way you kind of know that these lipoproteins, what these things look like for one, where the proteins are located in them, right? And knowing what we know about proteins, proteins can be polar or nonpolar, right? We haven't really had the protein chapter yet, which comes up, I believe, on chapter 13. But proteins have polar portions and they have nonpolar portions. And we know that because we've talked about the amide linkages, right, that link the amino acids together. We can see those carboxylic acid groups, and we call it amino acid, right, the amine group and the carboxylic acid group, and binding these things together through amidation reactions. So we know obviously they do have polar portions, and some of the amino acids are nonpolar and some of them are polar. And so what we would actually see here, and something like kind of important, I believe it's a question that I noticed on kind of the additional questions pamphlet or packet things that I uploaded earlier today. So the nonpolar amino acids would be here, and they would be interacting with the nonpolar tails of the lipids, and then the polar portions would be on the outside, interacting with the aqueous environment, and also kind of stuck here with the polar head groups and interacting with those. So really, part of the body and the organization of it is we put polar things with polar things and nonpolar things with nonpolar things, right? Like always dissolves like. And it would make sense that the outer part, or this part up here of this protein, would be polar, and the part down here would be nonpolar, right? So these lipoproteins, they're assembled, they're spherical shaped, and triglycerides and cholesterol are transported in the nonpolar core. So cholesterol is also nonpolar. Before we talk about cholesterol, though, we'll talk about just a few different types of lipids. We've discussed glycerol and triglycerides, but there are other forms or versions of lipids. And the other one that is extremely important to know is this sphingosine, or these sphingolipids. The big difference, right, kind of like a compare and contrast between a triglycerol and a say sphingosine, or like a triglyceride or a sphingosine lipid, is that in a triglyceride we have 3 fatty acids, right? This glycerol are esterified at each of these alcohol groups with the carboxylic acid that's on the fatty acid. Now in a sphingosine, part of the tail itself is the sphingosine backbone. And so we have this backbone portion, and actually part of the long carbon tail itself is part of sphingosine. And so it's kind of demonstrated here that this is one of the long carbon tails, and then we can attach a carboxylic acid or a fatty acid here through amidation, and then we can attach another fatty acid here through esterification. So instead of just being one glycerol, 3 fatty acids can be attached. In a sphingosine, we have part of the sphingosine itself is one of the long carbon chains, and then we have amidation and esterification right here. And so we call these molecules amphipathic. It means that they have a polar portion and a nonpolar portion. So it's important to know that term, amphipathic. So membrane lipids, they contain glycerol or sphingosine backbones. So membrane lipids are a combination of both. And what we'll learn in just a second is that we also have these phospholipid. So I know you've heard before the phospholipid bilayer. It's where they kind of face each other to form a bilayer instead of a monolayer, which would be what is right here. Here would be considered a monolayer. We just have one layer of lipids. In a bilayer, we have 2 layers because the inside of the cell is aqueous and the outside of the cell is aqueous. So they're composed primarily of phospholipids and glycolipids, with cholesterol adding rigidity. So cholesterol can be stuck in there in the nonpolar portion of the lipid bilayer and increase the rigidity by increasing the atoms that are there to interact through nonpolar forces like London dispersion forces. Well, glycolipids are just like what it sounds like. It's a glycolipid, so it's partially lipid, and it also has say like a glucose or another type of sugar that's attached to it. Membrane lipids, they contain glycerol or sphingosine as backbones. When the sphingosine is the backbone, one of the hydrocarbon tails comes from the sphingosine itself, kind of like I just explained. And so here's an example of a membrane lipid, or what we would call a phospholipid. So this would be a glycerol backbone. And when you see a phospholipid, you actually usually see the shorthand version, kind of if we look over on the right of the screen. Like here we have the three long hydrocarbon chains. Now when you see a phospholipid, or a phospholipid bilayer, it's usually drawn in shorthand with just 2 chains. And this is why. It's because when you look at it in depth and you actually look at the, this isn't the full Lewis dot structure, but like this part is condensed. But you can see the major linkages that we actually only have 2 of the nonpolar tails, and this is the whole polar head, which usually in the shorthand is just drawn as one full circle. So we have this phosphate part, we have the glycerol part, and then we have this thing called a choline, which would be considered an amine, right? And we can kind of see that it has a polar charge because this nitrogen has 5 things attached to it, right? And we kind of know what that means as far as like the valence shell portion, or the VSEPR theory. So for this slide, before I trim this thing down, it did include in-depth looks at each one of these. Really what's important to know is that this glycerol, or this triglyceride, we also call it a triacyl glyceride, which I think you probably heard me kind of slip up and say a few times in class. Both are the same, or both are correct, triacyl glyceride or triglyceride. This is mostly for energy storage, and we have the glycerol portion with the 3 fatty acid tails. This is what all this stuff on the right is really explaining, energy storage. Now membrane lipids, we have kind of an array of different lipids that we can use to build membranes. We have the glycerophospholipid, right? We have the glycerol portion, the phosphate portion, and the amino alcohol portion. And that's kind of like what this thing right here is, right? We have this amine, phosphate, and glycerol portion. We can also have what we call a sphingophospholipid. So it's a sphingosine backbone, kind of like we talked about a few slides ago. We have this one fatty acid, and then at the other place where the fatty acid would be attached, we have the phosphate and the amino alcohol. So if we were to look back here, we have the hydroxyl right here that forms the long fatty acid portion. And so one of the fatty acid, or one of the long carbon chains, comes from the sphingosine itself. The other is through esterification of a fatty acid. And then at this nitrogen portion, we have the connection of a phosphate. And so, there are a few different ways we can attach these things together. And then we have the glycosphingolipid. What that means is it has a sphingosine backbone, right? We have one fatty acid tail that's attached through either esterification or amidation. And then at the other one, we either have like a monosaccharide, a disaccharide, or even an oligosaccharide, where we have multiple sugar molecules attached. So this is kind of like what the shorthand looks like. We have the nonpolar tails and the polar head. Just know that this is representing, could be any of these things right here, right? Where we just have 2 tails here, 2 tails here, or 2 tails here. If it's sphingosine, one of the tails is part of the actual sphingosine backbone itself. These are either phospholipids or a glycolipid. It can either be a glycerol or a glycerolipid or a sphingolipid, depending upon which backbone is used, the sphingosine or the glycerol. And so this is why you see the 2 long tails instead of 3, which when you see 3, it is really mostly this triglyceride, which is used for energy storage. And so this cell membrane, it forms these 2 layers of a phospholipid and glycolipids assembled tail to tail in a bilayer, often called what we would consider a fluid mosaic. So this mosaic, really what that means is that these things are moving around. If you kind of imagine these things almost like animated, it's kind of like moving around, and this protein isn't stuck in this one location. It can travel across the cell surface and move around, and it's kind of things kind of bumping into each other, almost like you would imagine. I kind of like when I look at it, or when I've seen these types of animations of these things, it almost looks like a whole bunch of those, like a ball pit. But if you were to take all those little balls in that ball pit and put it into water, where it formed one big layer, and the water is kind of moving up and down, and you can kind of see these things floating around. That one of those balls isn't like stuck on say the right side of the pool, or if you were to put it into like a bathtub or something like that. After a while, that ball could move all the way over to the left side of the bathtub or pool or whatever you're imagining. So these things that's what we mean by when we call a fluid mosaic. It is fluid. It is not rigid. And so these carbohydrates that are kind of sticking off here, these would actually be part or connected to the lipid itself. And it would be one of these glycosphingolipids. So we can have an oligosaccharide that's attached. And these are actually used for cell surface markers, where we can identify cells, or cells identify each other based on these long carbohydrates. One example, right, is the A positive, B positive, AB positive, negative. Those blood types, right, are different forms of carbohydrates. So usually those are bound to the lipids themselves through these glycosphingolipids. As far as the diffusion and other things that have to do with the cell membrane, so it's important to know the lipid bilayer, right? So it is a bilayer, meaning that we have the polar head groups here and nonpolar tail groups here. Well, we have other phospholipids or other types of membrane lipids, any one of these three things right here, facing the other direction. And so this protein, kind of like I explained in the chylomicron, this protein right here has the nonpolar portion of the protein would be dissolved, or we would say in contact with the nonpolar tails, and then the polar portion would be on the top or the bottom because it's aqueous on the inside of the cell and it's aqueous on the extracellular side of the membrane. So it's aqueous on both portions. And so certain things, that means that only certain things can get through. So small nonpolar molecules pass freely through the membrane, through diffusion. Water passes slowly by osmosis and usually only through these specific channels. We call them aquaporins. It's a specific channel. I've always remembered that name just because it's like water is pouring into the cell or out of the cell. So aquaporin is a specific channel for water to go into or come out of the cell freely, usually through osmosis, but it takes a while because it's not able to diffuse across the entire cell membrane. So it makes sense why small molecules and nonpolar, so small nonpolar molecules would be able to pass freely through. It makes sense because the cell is very nonpolar on the inside. And so the inside of the cell right here is nonpolar. So if it's small enough to get past the polar head group and it is nonpolar, then it would be easy to dissolve into the nonpolar portion. Transport proteins facilitate the transport of ions from one side to the other. So if it's charged, it's not going to play nicely with the nonpolar portion of this lipid bilayer. Hormones, they regulate transport. Permeability depends upon saturation. So obviously when we talked about diffusion, right? Diffusion is based off of whether or not something is saturated on the outside or if there's more on one side than the other. So whether it's permeable, obviously would depend on the saturation of the fluid on the one side or the other. So now we can kind of shift to the last part, 12.4, which is talking about steroids and cholesterol and the transport of these things using lipoproteins such as chylomicrons, which are also used, that we've already introduced, are used to transport triglycerides and fatty acids through the blood, which is an extremely polar environment. So steroids, they have this characteristic ring system. They are lipids because they are only soluble in organic solvents, but they are structurally distinct, right? They don't have that polar head group. Really, the core structure is this. So I kind of have this right here. So she'll do this exercise. So it's got these 3 cyclohexanes and 1 cyclopentane. So hexane meaning 1, 2, 3, 4, 5, 6, and then the pentane meaning 1, 2, 3, 4, 5, right? So it's a pentane and 3 cyclohexanes. Now there can be double bonds in there. These can be benzene rings, but they don't have to be. So this is just the core structure of steroids, right? So steroids have this specific core structure. And you can see from this, they're extremely nonpolar. Unless we were to put a whole bunch of hydroxyls or other things around here, which some of them do, they are very nonpolar. So they have this nonpolar core structure. And we do call them lipids because they're soluble in organic solvents, meaning nonpolar solvents. So practice exercise 27. Prednisone, it reduces itching, swelling, redness, and allergic reactions. Um, Naproxen reduces pain, fever, and swelling. So based on these chemical structures of the two drugs, which one is a steroid? So it's very easy to kind of look at this and see these 1, 2, 3, 6-membered rings and this one 5-membered ring, and to notice that this obviously this prednisone is a steroid. Now, a lot of steroids, they have the ending ONE, and you'll find that when you get into like learning about the medications and the hormonal system of the body. So this ONE, so prednisone, right? We have cortisone. So there's a lot of different names for these things, but all of them kind of have this ending ONE when they're looking at steroids. And steroids have this core structure. Now, cholesterol is the structural precursor to steroid hormones. So when we ingest things with cholesterol, our body uses those to create hormones. It also uses them, vitamin D, and to create these bile salts, which we talked about form these emulsions. When we discussed over on the right side of the screen, we discussed this gallbladder producing these bile salts and forming this emulsion, right? So it actually turns out that the bile salts, they do have the precursor of cholesterol. So cholesterol, when we say precursor, we mean that cholesterol is used to produce these different things. So bile salts, they're emulsifying agents produced in the liver, stored in the gallbladder, and they break up globules of dietary fat. So this would be considered a bile salt, a cholate, right? We call it a salt because it has this negatively charged carboxylate on the right side of the molecule. So carboxylate, right, we would usually probably see like a sodium, like an Na plus over here, neutralizing or ionically bonding with that negatively charged oxygen. So a few things, just kind of like terms and things to know about these steroid hormones. Right, steroid hormones we already talked about are synthesized from cholesterol. Glucocorticoids produced in the adrenal cortex. And when we say adrenal, it means ad above renal, meaning the renal system, right? And the cortex meaning the outside. So you'll kind of notice as you go through medicine that a lot of these things are named kind of like out of common sense. We kind of name them based on their location or the core structure, what these things really look like, their function sometimes. So a glucocorticoid, exactly what it sounds like. It would be like a glucose or some sort of sugar with a corticoid, meaning the steroid hormone or the steroid, steroid backbone. So the steroid core structure. So a glucocorticoid, they're produced by the adrenal cortex, outer part of the adrenal glands that rest right above the kidneys. So adrenal, above renal system, above the kidneys. In the outer part, right, cortex, outer part, kind of like when we talk about the cortex of the brain, it's the outer part of the brain. So these hormones, they regulate glucose metabolism, they have anti-inflammatory and immunosuppressant, suppressing the immune system, this activity. They can be used to treat autoimmune conditions as well as allergies and asthma. So when we ingest glucocorticoids, a lot of allergic or allergy medicines are usually glucocorticoids, and they're used to kind of suppress the immune response to allergens. So cortisol or hydrocortisone, it is released in the response to stress. So you've probably always heard cortisol being one of the major stress hormones. It's released in response to stress, causes an increase in blood glucose, and facilitates fat and protein metabolism. So it gets our body going. So when we're stressed, it really kind of gets glucose into the blood. That way we can deal with that stress and mitigate it. So then we can kind of go back into this rest and digest phase. And so prednisone is structurally similar to cortisol. And we can kind of see the structural similarities, right? Both of these would be considered a steroid backbone, right? Both of them have the steroid backbone. Really the biggest difference right here is that we have like kind of this difference in the hydroxyl group that's kind of located over on the right side. Other things just kind of like memorization based. Mineralocorticoids regulate electrolyte balance in tissues. We've talked about electrolytes. We've talked about how we quantify them in their concentration, right? EQ and milli-EQ. They are electrolytes, really anything that would conduct electricity because they're charged ions dissolved in the blood or in some sort of liquid. Mineral corticoids, that's what they do. They regulate this balance. Aldosterone increases blood pressure, and sex hormones, progestin, estrogen, and androgens, they control the development of sexual characteristics in the fetus and during puberty. So at a certain age, these things get turned on, and they actually control the development of these sexual characteristics. So kind of like we talked about, I kind of introduced it back then. Cholesterol and triglycerides are transported by lipoproteins. So when we ingest cholesterol, they're also transported by these glycoproteins because they are very nonpolar and they're stuck in the middle with the triglycerides. They assemble as a spherical shape, right? Monolayer of phospholipids, triglycerides and cholesterol are transported in the nonpolar core. So kind of like basing off of that or going off of that, once they are delivered, their contents to the cell, they return to the liver. So they're kind of like kept in the liver or stored there. And once they grab those, either the fatty acids or the cholesterol, and they take them to the cell and they dump them off in the cell, right? We talked about the hydrolysis of those fatty acids, and they return to the liver. They're then degraded or reprocessed into what we call very low density lipoproteins. So really the density portion, what that really means is the density of either cholesterol or triglycerides that are inside those glycoproteins. So if they are very low density lipoproteins, then they don't have really any or much at all of like cholesterol or triglycerides in them. As they unload cholesterol and triglycerides, they become intermediate density lipoproteins, then low density lipoproteins. So high density lipoproteins, right, would go around and scavenge cholesterol from the bloodstream and return it to the liver. So when we say HDL and LDL, we've talked about the bad, which one is bad for you, right? So there are various different types and functions. We have these chylomicrons, then they can be processed into these very low density lipoproteins, and as they unload cholesterol and triglycerides, they become intermediate density and then low density. And so these high density lipoproteins are an indicator of cholesterol level in the blood. So when we go get our blood taken and we look at HDL and LDL, when we look at which one's bad, really we're looking at, okay, it's kind of like an indicator. It's just kind of used as a tool or to indicate how much cholesterol is in the blood. If cholesterol is in the blood and it's not properly scavenged, then we form clots. We form blood clots. Those blood clots can travel and cause rupture of the blood of vessels, either in the heart or in the brain, and that can cause severe damage. So, kind of like this last slide here, they're differentiated by their density, right? Density meaning how much is packed into them. The greater the lipid content, the less dense the lipoprotein. So I know that's kind of like counterintuitive, but the more lipid content that these things have, when we say lipid content, we mean like triglycerides, right? So the more the lipid content, the less dense the lipoprotein. And so basically the less lipid content in them, they become more and more dense, which I know kind of sounds like counterintuitive, but it really has to do with like the spacing out of the molecules and like the polar head groups. So the various different types, they have different functions, right? Bad cholesterol is this very low density lipoprotein. That makes sense, right? So if we have the low density lipoprotein or very low density lipoprotein, we would consider that bad cholesterol because they're not going around scavenging cholesterol from the blood. This very low density lipoprotein would also mean that they have a greater lipid content, which means that there's a lot of lipids in them. And so this good cholesterol, high density lipoprotein, if we take our blood and we see that we have this HDL and we have this high density lipoprotein, that's good because HDL or high density lipoprotein is going around the blood and scavenging up cholesterol and returning it to the liver. So it's keeping us from getting blood clots. If we another indicator, this high density lipoprotein means that we don't have a lot of fats in them. It's kind of like it used as an indicator, and that's really why, right? So that's like kind of the end of that chapter. I'll start on chapter 13 on Monday. We'll go through part of chapter 13. I think what I'll do is I'll probably like push chapter 13 and like a little bit of chapter 14 into one PowerPoint. I'll upload that this weekend, and we'll kind of go through that on Monday and Wednesday. Make sure you post on there so we can get this review session. It's been awesome having you guys this semester. I really look forward to these last couple of days that we have together. If you have any questions as you're studying, like please definitely email me. I'll try to have like a little bit more permanent situation of the car. My dad is actually on his way up here. I think we're gonna try to fix this car today and see if I can get it like at least drivable. So we're about to go try to work on that. So hopefully I'll have a more permanent situation with that, and I'll be able to stay at school longer. And I'll be there a little bit longer. That way, if you need to meet with me, just email me and let me know. That way I can kind of like plan, and I can make sure I've got a room available, and I've kind of come to a stopping point in either my grading or anything else that I'm doing. So I'll see all y'all on Monday, and please again just kind of let me know if you have any questions at all. Thanks again. See y'all later.